ENOVEO is a French company based in Lyon. We are developing and applying molecular biology tools and biotechnologies for environmental issues, such as remediation of polluted sites, water quality monitoring, and biosensors. Our team is composed of 2 doctors, 3 engineers specialized in environmental microbiology.
We are involved in the Love to Aid project, which is a project financed by the European Union in the framework of the FP7 Marie Curie. So our part in the project is to assess the impact of the pesticides on the microbial communities using high-throughput sequencing techniques like Illumina sequencing. And we are also involved in the second part, which is the development of a pesticide chip, which is a DNA microarray, which will allow a final user to assess the gene content of the soil, and which will help them adjust the amount of pesticides which will be applied on these soils to reduce the environmental impact of pesticides. The PCR is one of the most powerful techniques used in molecular biology. It's a technique that we use a lot at ENOVEO. This technique allows to amplify a really specific DNA sequence to a really high amount of copies. The DNA amplified this way can then be used for further experiments like sequencing or cloning. One of the flavors of the PCR is the qPCR, the quantitative PCR, and the idea is to add a fluorescent dye which will allow to follow the amplification process in the reaction, and then using this approach we can actually determine how many copies were at the beginning of a reaction. Upon receipt, the samples are first checked and then they are stored at -20 for conservation purpose. First, the samples are diluted. 2 purposes for that. The first is to dilute the DNA, to not introduce too much in the reaction mix. The second purpose is to dilute eventual PCR inhibitors. Second step is to prepare the reaction mix. For that, all components are mixed in one tube. The mix for the qPCR, the DNTPs and the primers. Then this is dispensed in the small reaction tubes. We are now adding the samples and the standards to the reaction tubes. Here is the last step. We put the tubes inside of a qPCR machine and we run the machine. So what happened in the reaction mix is that we are now amplifying at every cycle the DNA. And the more DNA will be produced, the more fluorescence we will be able to measure. We can see here the increase of fluorescence in one tube, which is directly linked to the quantity of DNA which is produced. Using this, we can actually determine a standard curve, which will be linking the proportionality between the number of copies present in our tube and the cycle at which the fluorescence will cross a specific line called the threshold cycle line. Then the idea is to use the standard curve to produce our sample, and here we have our samples. And we report the cross between with the threshold cycle line, and we can then determine on this curve which is the concentration we had at the start of the reaction. In this case, we had 65,000 copies per reaction. So now we have an absolute quantification of our number of copies, but we want to make sure also that we actually measured what we want to measure. So to determine this melting temperature, we just heat our tubes from 50 degrees to up to 95 degrees, and we measure the decrease of fluorescence. One way to put this decrease of fluorescence is to express it as a speed. And here we have this peak which corresponds to the melting temperature of our sample. The fact that they are all aligned here in one peak means that we have only one fragment with no variation. So like that, we can assess if our quantitative PCR is or is not specific. In this case, it is. So now we've seen a quick overview of the qPCR technique. What can we do with the data we acquire this way? We can follow a population, follow the increase and the decrease in response to any stimuli. And we can also, for example, follow the degradation genes specific to a compound and assess the potential to degrade this compound in the environment.